A clone is a population of the cells or organisms derived asexually from a single ancestor. Means from a single organism, the cells are produced by asexually. They are genetically identical to each other to their common ancestor. The produced organisms by a sexual method are identical to each other. Cloning means the production of the exact genetic replica copy of an individual. Cloning means production of identical copies that are similar to each other. They cannot be considered as offsprings but the simply copy of a given individual. As we said before, cloning means the copy of a same individual. Much work has been done on cloning in plants and microorganisms. Cloning is done in both plants and microorganisms. Many animals from a single genetically superior embryo can be produced. Many animals are produced from a single embryo. Many animals are produced from a single superior embryo. Still, there is no method of finding out which embryo are capable of cloning. We cannot find out which embryo can be used for the cloning. That means we cannot find the embryo which can produce the same identical copies. Means cloning. Transfer of whole nuclei or split embryos. Transfer of whole nucleus from a somatic cell of a superior donor to the enucleated egg that can be achieved using the following steps. Transfer of whole nucleus from the somatic cell. Somatic cell means except the reproductive cells. That means sperm and egg cell. Except the sperm and egg cells, the remaining cells are known as the somatic cells. From the somatic cell, we need to transfer the nucleus to the enucleated eggs. That means which do not have the nucleus. We need to transfer the somatic cell nucleus into the enucleated eggs. That means which do not have a nucleus eggs can be achieved by the following steps. Enucleation of unfertilized egg is achieved by the centrifuging cytoplasm B treated cells such as that a nuclei detached from the cells and palate at the bottom. Unfertilized egg is treated with the treated cells by using a centrifuging cytoplasm method. This should be treated with a treated cells by the process centrifuging cytoplasm so that the nucleus will detach from the egg. From this egg, the nucleus will detach when we proceed this process and the palate at the bottom. The nucleus was detached and the remaining part will be settled at the bottom of the tube, leaving the enucleated eggs in the supernatant. That means at the bottom of the tube. Next, Karyoplasts are similarly obtained from the blastula. As the same process, the karyoplast also obtained from the blastula stage of the developing embryo of the donor. Cardioplasts are derived from the donor or incubated with the enucleated eggs. The karyoplast obtained here are incubated with the enucleated eggs. That means we should not have the nucleus. We need to incubate both of these in the presence of a peg. There is a polyethylene glycol and fusion is achieved. If we incubate these both, they get fusion. So that after the fusion, That egg is transferred into the uterus of the surrogate mother. Techniques also have been developed where a superior developing embryo may be bisected into two parts using a surgical blades. We can divide the embryo by using the surgical blade by cutting the embryo. Each bisect part embryo may be separately transferred. Each of the embryo after cutting, each of the embryo should be transferred to the uterus of a surrogate mother for the further development. Dolly. First cloning process is done in an animal known as done in an animal known as a dolly. It is done in an institute known as Roslin Institute in Edinburgh, Scotland. They successfully cloned a sheep named Dolly. The cloned sheep name is Dolly. Dolly was born on 5th July 1996 and was first mammal to be cloned. This is the first mammal to be cloned. During the process of cloning, Dolly, a cell collected from the mammary gland of the female fin dorsal sheep. To get the Dolly, first we need to take the cell from the fin dorset sheep. From the fin dorset sheep containing the mammary glands, from the mammary glands we need to take the cells. Simultaneously, egg should be taken from the Scottish black ivy. Fin dorset sheep. From this, a cell should be taken, and from this, an egg should be taken. The egg taken from this, the nucleus should be removed from the egg taken by this. The nucleus was removed from the egg. Then the nucleus of the mammary glands. The nucleus from this egg should be removed, and the nucleus of this cell should be inserted into this, so that the new organism was produced, name known as Dolly. Dolly shows the characters of the fin dorset sheep because the nucleus was taken from the fin dorset sheep, not from the Scottish black ivy. So that the Dolly shows the characters of the dorset sheep because nucleus was taken from this. Dolly was a healthy clone from the fin dorset. She produced several offsprings. Dolly produced several offsprings after her own normally sexual means. Unfortunately, Dolly died on 14th February. Dolly died on the 14th February 2003 due to the certain lung diseases.